All right, let's start. So it's my great pleasure to introduce Kaitlin Casaletto, who is an absolute rock star in her field of research and a very inspiring and kind colleague. Kaitlin Casaletto is a neuropsychologist and associate professor at the UCSF Memory and Aging Center. She is passionate about understanding resilience to brain aging and neurodegeneration. She obtained her PhD in clinical psychology from the UCSD SDSU Joint Doctoral Program and her fellowship in clinical neuropsychology at the UCSF Memory and Aging Center, where she went on to join faculty. Her research program aims to prevent cognitive decline by identifying the biological and behavioral drivers, especially sex and genders, of cognitive resilience with age. Her work incorporates fluid biomarkers, neuropathology indicators, and digital health capture of behavior to deeply understand human brain aging and disease. Her exceptional research has been featured in the NYT, CNN, and NPR, including a novel blood-based protein associated with exercise and brain rejuvenation, and identification of synaptic and inflammatory biomarkers as targets of exercise and cognitive resilience. Kaitlin Casaletto, thank you so much for joining us today. The floor is yours.
Thank you, Nina, so much for the kind introduction, and just to the organizers for inviting me here today. So I'm just going to share my screen. Oops. Okay, does that look okay to everyone? Perfect. All right, so I'm just thrilled to be here to talk a little bit about sex differences across neurodegenerative diseases and what we've learned in terms of lessons of cognitive resilience. So first, I wanted to level set, what am I talking about when I say cognitive resilience? So I thought I'd walk through a couple cases. These are real cases we've seen in our research program. On the left-hand side, we have an 81-year-old woman with 20 years of education. On the right-hand side, a 74-year-old man with 20 years of education. And we have their T1 structural imaging in their FLAIR. And you can see this woman that her ventricles are enlarged, suggesting that there may be some global atrophy. You can see her sulci a little bit enlarged. And in particular, her hippocampi have a significant amount of atrophy. And if we look on her FLAIR here, you can see that there are white matter hyperintensities around the ventricles, confluent. There are also some punctate lesions across the brain, suggesting a moderate burden of cerebrovascular disease. And if we look at this gentleman over here on the right, though, his brain looks nice and plump. His hippocampi are full. On his FLAIR imaging, there's very little evidence of any white matter injury, just a little bit of capping of the ventricles here. But what do they look like when we start to test them? Well, this woman on the left is diagnosed as clinically normal. Her global cognitive screener, she scores a 29 out of 30. On a list learning test, she can recall 15 out of 16 words after a 20-minute delay. And she's really speedy on a trail making test. Whereas this gentleman on the right is diagnosed with mild cognitive impairment. He's impaired on his global cognitive screener. He can't recall any words on a list learning test after a 20-minute delay. And he's quite slowed on a trail making test. And so these are the cases that always pique my curiosity, make me so interested. And really the question is, what is the secret sauce that this woman has, and how do we all get some of that? Okay, so again, how do we wrap our head around these phenomenon that we observe of cognitive resilience? This is a working model that I've been working towards to try to operationalize some of these phenomenon. It's adapted from other working models that I recognize down here. And essentially, it splits this phenomenon into two constructs: resistance to the development of pathology and cognitive resilience in the face of pathology. And I think it's helpful to split into these two constructs because I think about resistance as primary prevention tools, ways of preventing the pathology from ever occurring, whereas cognitive resilience may be thought of more as secondary prevention, so preventing the morbidities that come along with the development of pathology. And let's just walk through an example to make this really concrete. A risk exposure could be something like a brain full of amyloid plaques, but perhaps that individual is showing resistance to the development of tau pathology. And so we might want to try and figure out, well, what is promoting this resistance? On the other hand, you could have someone with a brain full of tau pathology who's not manifesting any cognitive or functional symptoms of that pathology. So of course, we want to try and understand what's driving that. So ultimately, our goal in our lab is to fill in these boxes. And we think if we can figure out the biological systems, pathways, and ultimately the individual molecules that drive resistance to pathology and cognitive resilience, I mean, these are our golden tickets. This is how we all get to age well in terms of brain aging and how we can prevent dementia. But obviously, it's not that easy. It's really hard to find these molecules. Otherwise, we would have done it. So what our team does is we kind of take a step back and think about, well, what are some human factors that we already know are neuroprotective based on other work that are associated with these resistance and resilience phenomenon? And might we dig very deeply into these neuroprotective factors in order to identify some of these resistance and resilience molecules? And so our team is really honed in on factors like physical exercise, which we know promotes neurogenesis, cardiovascular health, cognitive stimulation, and really dug into these phenomenon. But we're here to talk about sex. And so this story is one really of following the data. So how might we think about sex in the context of resilience? So we were, looking at these data, publishing papers again and again on cognitive resilience, and we are, good followers of Francis Collins and would look at sex as a biological variable. And time after time, after time, after time, after time again, we found that there were major sex differences in our analyses. And not only that, but sex was the headliner. It was the cool part of the story. And it sort of had this moment of like, of course, this is the case. There are fundamental biologies that differ between men and women, not to mention the social political construct in which they function. And so I just became really intrigued by trying to understand this a bit more deeply. So what if we think about this model and revise it slightly to think about sex as one of these potentially neuroprotective factors? What if we dig deeply here? And the more I looked into this, the more compelling it became. So perhaps the female body can be used as a model of resilience. Why? Well, women seem to have a sensory advantage. So women have better olfaction and better hearing than men. These are some quotes pulled from abstracts that I think just tell the whole story. Women also have a metabolic advantage. This is particularly true for ultra endurance capacity. So women seem to have this ability to shift their mitochondria into this lipid oxidization benefit, which essentially also promotes resistance to the development of oxidation deficits like insulin resistance. And what I think is sort of the ultimate outcome, women have outlived men. They have a survival advantage. This is across countries, cultures, and species. So these advantages are not huge, they're small, but they're biologically robust, which suggests that there's an evolutionary basis deep in our roots, I think, to these advantages. And when we think about the brain, like, my gosh, the primary sensory information coming in to stimulate our brains is maybe better in women. Our brain, which is the most metabolically demanding organ in our body, and living longer. So I think there's really an interesting story that we can start to understand using sex as a model of resilience. And for the early career folks out there, why study this sex and gender differences? Well, there's a major gap here. And what this translates into in scientific terms is huge opportunity. So this is a plot developed by Emily Jacobs and her team that I'm sure many of you have seen, that essentially plots the number of publications over the past 25 years using neuroimaging. And you can see this really incredible blue wave of publications on neuroimaging over the last 25 years. And if you look at the yellow wave here, you can see that these are all the articles that were studying depression and neuroimaging. And if you, squint quite deeply, you can see there's a small black line here. And those are the papers that represent neuroimaging, and looking at women's health factors. These are things like menstrual cycle, pregnancy, menopause. Ultimately, less than 0.5% of brain imaging articles published over the last 25 years have considered health factors specific to women. Half of our population is experiencing these health factors. And, I would argue it's the half that's doing 3/4 of the unpaid work in our societies. So when women's bodies and women's brains are well, communities thrive, societies thrive. I think we are incredibly well poised now to make some major scientific breakthroughs simply by digging into health factors that are specific to the female body. This is a really exciting time. Okay, so what are we going to do today? I mean, this is really the throughput question of the rest of the talk is, can we leverage biological sex to unlock healthful aging for all brains? And I'm going to touch on some studies ranging from typical cognitive aging across neurodegenerative diseases. I'm going to warn you that I only have time to really touch briefly on each. And for me, I have more questions than answers, and hopefully that's the case for you as well by the end of this talk, and inspiring new lines of thinking. Okay, so to start with, I talked about some of the other advantages outside of the brain that women have, but there's also this really interesting phenomenon that women appear to have better verbal memory than men. And this is, again, a very consistent phenomenon. We see this in children, including prepubescent children. This is on a list learning test. You can see that girls are simply performing better than boys across the learning trials and after delays. We see this in adults, females performing better on these list learning tests. We see this in disease. This is in the ADNI cohort using a different list learning test. You can see women, again, outperforming men. And we see this in postmenopausal women. This was a longitudinal study in our healthy aging cohort, and we looked at people who were more susceptible among normal people to show memory decline, and male sex came up as one of the key factors. So this is really fascinating. So women have a verbal memory advantage. What is the biology driving this? And if we could start to understand that biology, could this be used to treat memory disorders for all brains? So where do we start here? We thought we would sort of start at the beginning, which as a neuropsychologist is the synapse, just to start to untangle this question. So why the synapse? Again, as a neuropsychologist, I think of synaptic functioning as the biological correlate of memory and cognition. Interestingly, synaptic functioning tracks most closely with cognition, more closely than cell loss or pathology. And synaptic functioning seems to be particularly preserved in cases of cognitive resilience. So the integrity of the synapse, the molecular representation of the synapse, and even synaptic genes seem to be overrepresented in these cognitively resilient cases. Essentially, if you have a working synapse, it doesn't matter all the other crap that's happening in the brain, you can still function. And so this is where we wanted to start to understand what might be happening. And one protein in particular that we're going to dig into is called SNAP25. This is a presynaptic protein that's really critical in the snare complex, which helps release neurotransmitters into the presynaptic cleft. It's dysregulated, we know, in aging and Alzheimer's disease. And it also has hormone-dependent regulation, which made it a really interesting target for this sex question. Interestingly, there's a SNAP25 genetic SNP that's linked to neurodevelopmental disorders and young adult cognition, but it hadn't been looked at in older adults or in disease. And so this was exactly what we aim to do. And this was a study led by Rowan Sloaner, who's new faculty here at the Memory and Aging Center. And he wanted to look at this synaptic gene, SNAP25 SNP, on cognitive aging. And so this is what we did. We took this SNP within the SNAP25 gene. Essentially, there's a very common polymorphism where there's a T to C mutation that leads to less microRNA binding and essentially greater SNAP25 protein expression. And we sequenced 2 cohorts of older adults here at the MAC on 2 different arrays. So we had a discovery cohort and then a replication cohort. And then folks completed our typical neurobehavioral outcomes, so cognitive testing in both cohorts, brain MRI and amyloid PET, and then we simply asked questions about sex and this polymorphism. So what did we find? And in some, essentially the SNAP25 gene related to verbal memory more strongly in women, but not in men. So you can see that the C carrier, the female C carriers, show this verbal memory benefit, or is associated with a verbal memory benefit, not observed in the T carriers. And this benefit is not seen in men. Interestingly, this did replicate in our independent cohort as well. And if we start to look at brain structure, we can see that the female T carriers also had larger brain volumes in Alzheimer's disease related areas. So this was an Alzheimer's region of interest of gray matter integrity. And you can see again, C carriers, better structural integrity of these regions. This was sort of trending in the men as well, but you can see that the effect size is much smaller and it didn't reach statistical significance. And there was a functional consequence. So essentially, the female C carriers, those that had larger brain volumes, strongly correlated with their verbal memory performance, suggesting that in the C carriers, there's a particularly strong relationship between this fortified brain structure and verbal memory. This relationship was attenuated in the T carriers across sexes. And then really compelling, we had amyloid PET scans on some individuals. And again, we can see that the female C carriers had significantly lower rates of amyloid PET positivity compared to the T carriers. And we simply didn't see that effect in men. And in terms of effect sizes, the effect was actually quite large. It was about an 80% reduced risk of being amyloid positive among the C carriers. And so just a really quick snapshot of data, but perhaps this SNAP25 SNP is beginning to account for the female memory advantage. Perhaps it's these women who carry this common polymorphism that's driving some of this verbal memory advantage that we're seeing. Importantly, maybe SNAP25 SNP could be a resistance factor to the development of amyloid accumulation in women and the downstream consequences of that on brain structure and function. And then more broadly, stepping back, perhaps SNAP25 could be the protein or the gene, could be a neuroprotective target across sexes. What can we learn again from digging into a female-specific phenomenon that we can now apply across brains? And this is a target that we're very interested in and we can increasingly measure now. So we'll see where this data goes. Okay, so I just went on and on about how women have this verbal memory advantage, but there's a bit of a paradox. So despite having this verbal memory advantage, 2 out of 3 Alzheimer's patients, a disease that its sort of hallmark is memory deficits, are women. So women are, significantly disproportionately represented in Alzheimer's patients. What is going on here? If we look at the pathology, though, a story starts to emerge. And so this has been data that's been shown in cerebral spinal fluid, PET imaging, and neuropathology outcomes that women seem to carry a higher tau burden for their amyloid levels. So this is one of the seminal studies that was led by Rachel Buckley in 2019 using PET imaging. And this is in clinically normal men and women. And this was on the x-axis here, amyloid levels, and on the y-axis, tau levels. And you can see that women show disproportionately higher tau levels for their amyloid burden. We also observed this in our local cohort in our ADRC. So this is in impaired patients who have MCI and dementia. PET imaging again, amyloid on the X, tau on the Y, women showing, again, more tau for their amyloid burden. Okay, so maybe this is starting to account for what we're seeing in terms of the representation of women with Alzheimer's disease. However, if we look at these women, we can't tell that they have more tau. So these are the clinical data from this Edwards study. And you can look at the men and the women across all of these functional outcomes and memory outcomes, and there's absolutely no difference. So women are carrying this higher tau burden, but we're not seeing the symptomology of it. To me, this is sort of a classic resilience paradigm. And I think what makes it even more compelling is when we look at that ultimate outcome of survival. This is a recent meta-analysis looking at factors that are associated with survival in patients with Alzheimer's disease. And you can see that male sex was one of the strongest effects of faster time to death. This effect size is comparable to someone having cancer or having a movement disorder. So again, really fascinating and what is happening here. Why are women living longer and outperforming their tau levels? So I just want to take a minute to look a little bit more deeply at these data. These are some plots that were developed by Jessica Caldwell. This pattern of findings has also been replicated by Aaron Sunderman's group. So we're just going to take a minute and take in these plots because I think they're just really brilliant. So here on the y-axis, we have a memory test, so learning performances, delayed recall performances, and on the bottom, we have hippocampal volumes left and right. Each panel, we have men and women. On the x-axis, we have amyloid positivity, so negative or positive, and then the lines represent the diagnostic stage, so normal or early mild cognitive impairment. So let's just hone in on the blue lines for a minute here. So we can see among cognitively normal men and women, women again show this verbal memory advantage. They are higher than men. And interestingly, once they become amyloid positive, they don't show as strong of a cognitive decline as men are showing. And we can see this across learning, across delayed recall. We also see it in hippocampal volumes. So they're sort of resisting or being resilient against these amyloid conversions. And then if we shift now to the MCI plots, however, we can see that once women become positive, they actually show a steeper, once women began to manifest clinically their disease as mild cognitive impairment, they show a steeper effect of having amyloid, much steeper than in men. And we see this again on learning, we see this on memory. To a lesser extent, we see it a little bit on hippocampal decline as well. But I will say the other interesting point of these plots is even though women are showing a steeper decline in the MCI stage, where they land is still significantly higher than where the men are landing. So they're starting out a little bit higher and landing a little bit lower. And when I look at these plots, they just hearken some of these really classic plots of cognitive reserve. These are plots that I've seen from Jakob Stern and other leaders in the reserve field, where essentially any factor that is associated with high reserve is thought to sort of start someone out higher on that outcome and allow them to hold on longer despite accumulating pathology. However, once they hit their inflection point, we see a steeper decline after holding on to that pathology compared to someone with low reserve who starts out lower and just has a more gentle decline overall. And so this is a curve that is seen with education, so holding on longer with a steeper decline. And, you know, I would argue if you look closely and simply take these lines and plot them over here, that we're seeing a very similar curve between men and women. So I think what we're starting to see in women with Alzheimer's disease is a little bit of this cognitive resilience phenomenon. I think there's a lot to unpack here, and I'm really excited to dig into these data further. I don't have too much more of an explanation biologically for this yet, but I think this is a really exciting space. So we wanted to start to understand the sex differences in AD a little bit. So we actually decided to start a little bit earlier in the disease. So why do women have more tau? And in order to answer this question, we turn to the immune system because we know that the immune system has fundamental sex differences between men and women. And so perhaps this could be one of the keys to understanding why women, are accumulating more tau. So what do we know? Well, the X chromosome contains the largest number of immune genes of the whole genome. So I think that's pretty telling. Peripherally, we see that women have more robust and faster immune reactivity, faster wound healing, including faster recovery from TBIs. And after a vaccine, women show greater antibody production. So really robust immune system, which of course, on the other side of the coin, comes with some other downsides. About 80% of autoimmune diseases are in women. In the central nervous system, I think we're still learning a little bit more, but there's some data that suggests that women might have a heightened disease-associated microglial response to amyloid, which could result in poorer phagocytosis by the microglia and less compaction and coverage of amyloid plaques by microglia. And as a reminder, microglia are the key innate immune cells of our brain. And so essentially, we wanted to ask this question, well, might microglial activation help explain some of the sex differences that we're seeing between amyloid and tau? And so this was a project with the analytics really driven by my friend and colleague, Emma Nichols, who is now a research scientist at USC. So to address this question, we turned to the Rush Memory and Aging Project. This is a study of people who have donated their brains to autopsy. And you can see this is what the cohort looks like. I'll just point out that men were more highly educated than women. Men had slightly higher rates of mild cognitive impairment and dementia, although this did not reach statistical significance. But there were no differences in their proportion of activated microglia. And essentially what we measured from brain tissue was amyloid plaques and tau tangles using silver staining, and then microglial activation using immunohistochemistry, tagging different HLA isoforms, and then the morphology of the immune cells themselves. And we created, or we used this composite score that was created by the Felsky paper. And essentially it gets at the proportion of stage 3 activated microglia. So these are thought to be the most detrimental, pro-inflammatory forms of microglia. And so what did we find? So again, essentially our model was, does microglial activation mediate the relationship between amyloid and tau, and does it do it differently between sexes? And so for mediation, we're really looking at this indirect effect as the mediational effect. And what you can see here is that in women, about 57% of the relationship between amyloid and tau was explained through microglial activation. Whereas in men, only about 19% of the relationship, and this did not reach statistical significance. And we had this really beautiful double dissociation with the direct effect. And the direct effect is essentially the relationship between amyloid and tau, not explained by microglial activation. And you can see in men, it was the vast majority of the relationship, not explained by microglial activation. So perhaps microglial activation is having a particular role in how amyloid converts to tau in women that we're simply just not seeing in men. But of course, these are autopsy data, observational, and so we wanted to test another potential model. Might it be that microglial activation comes on board first and then leads to the amyloid and tau cascade? And so when testing this model, interestingly, we found that in women, it was about actually half and half. So about half of the relationship between microglial activation and tau was explained by amyloid, but the other half was just simply a direct effect. And both of these pathways were significant in women. Whereas in men, again, we only see that the direct effect is the primary pathway. So just simply microglial activation to tau, not explained through amyloid. And so if we take these data together, might this suggest that microglial activation or homeostasis might be a pathway to tau resistance that we're learning from men? So in women, we're seeing this bidirectional feedback loop between microglial activation and amyloid to tau that may be accounting for some of this disproportionate tau burden, whereas in men, it's a simple 1-to-1, more amyloid, more tau, more microglial activation, more tau. So sort of a fundamental difference, perhaps, in how the immune system is reacting to these proteinopathies. And if we take this now together, thinking about Alzheimer's disease, so we see that for their amyloid burden, women carry a higher amount of tau than men, and perhaps this can be explained in part through microglial activation. And in terms of the clinical phenomenon, I would argue that women are showing a little bit more of this cognitive resilience pattern, such that they start out with a higher advantage in terms of memory and survival. They hold on for a longer time with their amyloid and tau burden, but they show a more precipitous decline once they begin to manifest symptoms compared to men. So we've looked deeply at Alzheimer's disease and cognitive aging. How do we take these patterns and look across neurodegenerative diseases now and try to start to see, might there be any through lines that we can think about? So I'm going to take just a quick minute to give a primer on frontotemporal dementia, as I'm sure folks are a little bit less familiar with this form of neurodegenerative disease. So FTD is an umbrella term that refers to a whole host of clinical syndromes listed here. They range from sort of the classic behavioral variant frontotemporal dementia, which results in changes to personality and empathy and social and emotional skills. There's also language forms, primary progressive aphasias. There's a motor neuron disease, or ALS, form of FTD, as well as primary motor syndromes, cortical basal syndrome, and progressive supranuclear palsy. And all of these syndromes are caused by some also heterogeneous pathology. So there's FTD tau, which is a different isoform than Alzheimer's tau. And then there's TDP43. And these data actually represent the brain banks here at the Memory and Aging Center. So the colors that are mapped onto the syndromes represent which pathologies are most commonly represented in those syndromes. And I'm not going to get too much into FUS and CHIP2D, but I think a really interesting thing about FTD is it's fairly familial. So about 40% of people with FTD have a family history. And in about 20% of cases, we can identify a single autosomal dominant gene that causes FTD. This is much different than in Alzheimer's disease, where the autosomal dominant forms of AD are only representing about 1% of AD cases. And so I think it's a real opportunity to understand how a disease manifests when there's an autosomal dominant model. And so the genes that I'm talking about, there's 3 that are the most common. There's progranulin, C9 open reading frame 72, also represented here. And then MAPT, which causes tau. So MAPT for tau, progranulin, and C9 for TDP43. Okay, and what's been done in terms of sex differences in FTD? Not much, unfortunately. There's a couple papers here and there. I will highlight there's this really beautiful paper from a colleague, Ignacio Langala, a couple of years back, where he essentially argues that he's seeing female behavioral and cognitive resilience in behavioral variant FTD. So again, this is actually the most common form of FTD. So he honed in on this one syndrome, and he found that women seem to appear clinically comparable across cognitive and psychiatric outcomes, despite greater brain atrophy. And so these are heat maps representing the brain atrophy of the BVFTD men compared to controls and BVFTD women compared to controls. And, just looking across these two, I'm sure you can appreciate that the heat maps are much hotter for the women, greater atrophy. And if you look at the interaction, these are the regions that pop out as statistically significantly different. And you can also quantify this difference between brain atrophy to clinical outcome as a residual. So essentially, the residual is how much better someone is performing based on their brain structure. And I'm just showing one here, but essentially he did this for executive functioning performances and found that women demonstrated significantly better executive functioning performances for their brain atrophy rates. So So really kind of compelling idea that perhaps there's this resilience phenomenon happening in BV-FTD, but this is just one of the clinical syndromes. It's cross-sectional, just a start, tip of the iceberg. And so we wanted to try to address this a little bit more comprehensively. This is work led by a data analyst on the team, Molly Memel, and we turned to the All-FTD Consortium. So this is a multi-site consortium following families who have autosomal dominant forms of FTD across North America. And again, the perk of following people who carry these genetic mutations is you can map out the natural course of FTD before symptoms even start, all the way through to the dementia stages. And this is what our cohort looks like. You can see the top 3 genes represented here. The men and women were quite matched across factors. I'll just point out that about half of the cohort is asymptomatic, meaning that they carry one of these genetic mutations, but have not yet manifested disease. And then the one area that differed, however, was a blood marker, a plasma biomarker called neurofilament light chain, or NFL. This is a marker of axon degeneration. You can think of it as a marker of neurodegeneration broadly. It's actually the best molecular marker we have of FTD pathology burden at this point. We don't have biomarkers for FTD pathology. And you can see that women had a significantly higher level of this neurodegeneration biomarker than men just at baseline. And I'll dig into that a little bit in a bit. So these participants were followed over time up to 6 years. They completed cognitive testing, functional measures with the clinical dementia rating scale, and then their plasma NFL over time. And to cut to the chase, what did we find? So we found that plasma NFL showed steeper increases in women compared to men, but we did not see any sex differences in cognitive or functional trajectories when not considering disease stage. So a really similar pattern, more atrophy, more neurodegeneration without that sort of comparable clinical decline. But once you start to look at disease stage, I think the story gets a little bit more interesting. So we found that women carrying these autosomal dominant mutations for FTD show steeper progression after symptom onset. So let's walk through these plots. On the x-axis here, you have years since baseline, and on the y-axis, we have cognition, functional decline, and NFL, or neurodegeneration. The solid lines are female, dotted lines are males, and then the colors represent what disease stage the participants were at baseline. And so if we just hone in on the blue lines, the asymptomatic participants, you can see there's no sex differences across any of the 3 outcomes. However, once we started getting into the mild behavioral and cognitive impairment, and especially dementia, women start to depart. So women are declining more quickly across cognition, functional decline, and their NFL levels are just exploding. So once they turn into this disease state, more rapid decline, but before that, no differences. I hope you're remembering the figure that I showed for Alzheimer's disease. This is sort of like an eerily similar pattern that we're seeing that is really, I think, kind of exciting to think about together. And if we dig into this a little bit more closely and think about their pathology burden, entering the study, we found another interesting pattern. So in these plots here, again, we have years since baseline on the X, and here we just have cognition across all 3 plots. But this time, the colors represent the participants' baseline NFL levels, or their baseline pathology levels, you can think of. So in red is high pathology, and blue is low pathology. And you can see in the asymptomatic stages that participants with low NFL don't differ, but those with high NFL, men are declining more rapidly than women. But this pattern does not hold once we get into the MCI and dementia stages. We don't see any sex differences in NFL here. But NFL is more strongly predicting cognitive decline in asymptomatic men compared to women. And we also see this for functional decline. Again, men with high NFL and asymptomatic stages showing more rapid impairments compared to women. Women in other words, that women are outperforming their NFL burden during the asymptomatic phases, but this does not hold once we get into MCI and dementia. So I think these data hopefully convince us that FTD manifestation does differ by sex. We're seeing higher and steeper neurodegeneration burden in women compared to men, both at baseline and longitudinally. And we see steeper clinical declines and NFL accumulation, particularly after symptom onset, this explosion of where the disease goes. To me, again, this harkens this pattern of cognitive resilience in women, where we might have this prolonged asymptomatic phase followed by faster declines. So it, again, really reminds me of this plot that we were showing that hopefully I've convinced you is reminiscent of Alzheimer's disease plot as well. And more pragmatically, though, I think it's important to think about if NFL is the best biomarker we have for FTD right now, we're seeing that NFL does not track as closely with clinical cognitive decline in women who are asymptomatic compared to men. So perhaps this biomarker might not be as sensitive a prognostic biomarker in women, especially early in the disease. So really important to think about when it comes to diagnosis and prognostic planning. Okay, so we've looked at Alzheimer's disease, we've looked at frontotemporal dementia, totally different proteinopathies, really similar patterns. What else can we learn? What other diseases might we look at? What about Lewy body disease? This was a project that's led by a very talented graduate student, Madeline Wood, and in collaboration with Jenny Rabin. You know, what can we learn from a totally different type of proteinopathy in terms of sex differences? So again, I'll give just a really quick primer on Lewy body disorders for those who are less familiar. In Lewy body disease, instead of the amyloid plaques and tau tangles, we have alpha-synuclein, and essentially these alpha-synuclein oligomers aggregate into these Lewy bodies, and these Lewy bodies are intracellular. They mess up how the neuron functions and eventually kill the neuron. And these are the hallmark of Lewy body disorders. And clinically, this disease can manifest sort of across the spectrum of Parkinson's disease all the way to Lewy body dementia. As with everything in the brain, it's all about real estate. So it just depends on where the Lewy bodies aggregate. If it's more brainstem and subcortical, you'll see more of the motor symptoms. If the Lewy bodies aggregate more cortically, however, you'll see more of the cognitive and behavioral symptoms. The other thing about Lewy body is it's highly common. It's one of the top 3 pathologies that we die with among people who live the oldest, and it's extremely comorbid with AD pathology. It has this really interesting, I think, phenomenon between how plaques, tangles, and Lewy bodies intersect. This is in individuals with sporadic forms of Alzheimer's disease, either sporadic early age of onset or late age of onset Alzheimer's disease, and you can see that over 40% of participants also died with Lewy body pathology. So just this really common co-pathology. So we see differences in AD and in Lewy body. And I think the really interesting thing about Lewy body is it can be a really nice model, a juxtaposition against AD, where in AD, we see that it's 2/3 represented by women. Parkinson's disease is the exact opposite, 2/3 represented by men, which I think, again, provides a really nice model to start to understand sex differences in these neurodegenerative diseases. So this was essentially the question: how does sex impact Lewy body pathology and Lewy body related cognitive changes, particularly in the context of Alzheimer's pathology, given the high comorbidity? We had 2 cohorts, 2 really large cohorts to look at this with. So everyone went to autopsy. This is the Rush group, the Ross-Map-Mars, as well as the NAC, the National Alzheimer's Coordinating Center autopsy data. And essentially what we looked at was Lewy body disease, neocortical and limbic versus none, amyloid and tau, using different markers across cohorts. And what did we find? So we found that women are significantly less likely to have Lewy body pathology when they die compared to men. This has been reported before. but helpful to see. And as we've also seen before, women have had in both cohorts greater amount of amyloid and tau burden, even adjusting for Lewy body pathology. So again, men have more Lewy body, women have more AD. This has been reported before, but nice to see across both cohorts, we're seeing these patterns recapitulate. More interestingly, when we started to model in both pathologies, we found that Lewy body associated with disproportionately higher tau in women compared to men. And so this is in the ROS-MAP Mars cohort. You can see that women with Lewy body pathology had significantly higher tau burden compared to men, or even compared to women without Lewy body pathology. We also observed this in the NAC cohort. So again, women with Lewy body pathology were significantly more likely to be in the later Braak stages. We did not see this effect on amyloid. So if you sort of replaced the tau tangles with amyloid burden, sex did not moderate the effect of Lewy body pathology on amyloid. This seems to be a tau-specific phenomenon. And if we start to look at women's health factors, we found that women with earlier age of menopause showed this exacerbated association between Lewy body pathology and tau burden. So this is just stratified by age at menopause. The typical age of menopause in the US is 51. And you can see that women with earlier ages of menopause, that having Lewy body was associated with significantly higher tau burden. And there was a nice dose-dependent response, lower in people with average menopause. And then we saw no difference in tau burden in people who have later ages of menopause. So perhaps sex hormones may be playing a role here and maybe protecting against tau accumulation, even in the presence of Lewy body among women with later age of menopause. And if we look at the clinical outcomes, we found that Lewy body pathology was more strongly associated with cognitive decline in men compared to women. So this is the ROS-MAP Mars cohort looking at different cognitive domains. And you can see that in men, having Lewy body pathology was associated with a significantly steeper decline in memory function that we simply just didn't see in women. It only reached significance for memory, but you can see there's a pattern here across the cognitive domains. We saw something similar in the NAC cohort as well, although for the NAC cohort, only the attention processing speed domain reached statistical significance. So perhaps men are showing greater cognitive vulnerability in the presence of Lewy body pathology. But if we dig into some of the female reproductive health factors, the story gets a little bit more interesting. So this is just looking at women in the ROS-MAP Mars cohort. And Madeline found that women with earlier ages of menopause had a significantly worse memory outcome in the context of Lewy body pathology. So younger age of menopause, Lewy body seems to have a stronger effect on memory outcomes. But as you might remember from a couple slides back, we wondered, well, Lewy body pathology seems to be relating to stronger tau outcomes in women. Might that be what's driving some of these outcomes? And in fact, that's exactly what she found. Essentially, the tau burden explained about a third of the relationship between Lewy body pathology and memory decline among women at younger ages of menopause. So essentially, what we're seeing in Lewy body disease is that women are showing greater resistance to the development of alpha-synuclein Lewy bodies and greater cognitive resilience in the face of Lewy bodies specifically. However, are all roads leading to tau in women? So more Lewy body pathology in women seems to be promoting tau aggregation, or is associated with greater tau aggregation in women. And in fact, tau might better explain some of the clinical manifestations of disease in women with Lewy body. It seems to be that women may not be manifesting alpha-synuclein, but instead might be manifesting tau when these diseases come on board in women. And interestingly, perhaps shortened hormone exposure is exacerbating these Lewy body-related tau and cognitive deficits in women. And I think this is a story that's also playing out in Alzheimer's disease. This is a a recent paper by Gillian Gofflin from Rachel Buckley's team showing essentially that earlier age of menopause, so this pre-menopause orange line associates with higher tau burden in women. So this hormone exposure may be really important in how tau is aggregating in women's brains. All right, I just showed you a ton of data. I'm hoping to leave time for questions, so hopefully I can summarize this briefly for everyone. Coming back to our model, what we can learn from sex to guide us in terms of resistance and resilience phenomenon. So I think what we're starting to see is that there's, at least in typical cognitive aging, there's a subset of women who carry this SNAP25 polymorphism that are showing resistance to the development of amyloid and some of the downstream consequences on brain structure and function. And perhaps this is a target that could be pursued for amyloid resistance. Women are also showing resistance to Lewy body pathology, and I think this is just fascinating, and mechanisms are unknown. Men, on the other hand, seem to show greater resistance to the development of Alzheimer's-related tau. Our data suggests that perhaps microglial homeostasis may be a key factor in this. How are men's microglia functioning in a way that could be protective against tau, and how can we leverage this to help everyone against protecting against Alzheimer's tau? And men also seem to be showing less FTD-related neurodegeneration. So this is FTD tau and FTD TDP43. Men have less brain atrophy, less of that NFL biomarker compared to women, mechanism unknown. Okay, and if we get to the cognitive resilience side, what are we seeing? So we're seeing that women seem to show greater cognitive resilience against Lewy body pathology specifically on their cognitive outcomes. Maybe sex hormones are playing a role here in terms of promoting that resilience against Lewy body pathology. And this is, I know, a little bit of a controversial take, but I'd say for Alzheimer's related tau, that women are also showing cognitive resilience against their high burdens of AD tau. We see this for cognition and for survival. I think the mechanisms here are totally unknown, but incredibly exciting to think about. And we're seeing a very similar pattern with FTD, greater cognitive resilience to the neurodegenerative burden that women are carrying for cognition and function. Again, mechanism unknown. So as promised, more question marks than answers here, but just taking a quick step back, sort of a meta analysis of our plots so far, I will point out that there seems to be a pattern in our cognitive resilience box. Might there be female-specific pathways that are a throughput across these proteinopathies, and how can we leverage the female brain to start to understand these? So those are really fun, I think, intellectual gymnastics to put ourselves through, and this is why we do the work that we do. But more pragmatically, there's incredibly important real-world implications to these data. So even if you don't agree with my hypotheses or my conceptualizations, I hope you take this one take-home point, that there are fundamental differences in the development of and response to neurodegenerative proteinopathies by sex. What does this mean in terms of clinical care? This means that biomarkers of disease may be changing earlier in women compared to men. This is actually kind of a good thing in many ways. Perhaps we could catch women earlier in the disease process, a real opportunity for intervention. But the reality is it's so hard to catch someone when they're not manifesting diseases. And so how do we wrap our heads around making sure to get to the early intervention for women if they're not clinically manifesting these diseases in the same way as men? In terms of clinical care, I think that the data that suggests that women might have a more rapid trajectory after symptom manifestation means that they might need additional support and additional monitoring in clinic. And so being mindful of that as clinicians may be a key part. And then counseling. Very few people in our clinic are counseling our patients around how sex can influence disease trajectory. This is the first question that every family comes to us with. So I think that we can start to understand sex and health factors that track with sex to help families plan advanced directives and care for their loved one. And in terms of clinical trials, my gosh, are we enrolling women too late? Again, if we're enrolling them when they're manifesting disease, this may mean that they're carrying a higher neurodegeneration burden. Unfortunately, this has really played out in lecanemab, which is one of the very few disease-modifying therapies that we have in clinic right now in Alzheimer's disease. So the clinical trial essentially showed that there's less than half benefit in women compared to men. I mean, this is a disease that hits women much harder than men. This is just a huge disappointment in terms of treating the people that need to be treated. And I think we can learn from this moving forward. When we start trials for FTD and Lewy body and additional trials for AD, I would argue that we need powered study designs that are able to be stratified by sex. This is sort of necessary. It's not optional if we truly believe that these diseases are manifesting differently across sex. All right, next steps. These are the fun parts of our job. The 3-year-old gets to come out and ask, why is this happening? How is this happening? So the areas that we really want to tackle are trying to understand the sex chromosomes, so the X and the Y chromosome. What can these biologies teach us about brain aging, resistance, and resilience? Of course, sex hormones is the other major biology that differs by sex. I think really deeply understanding female reproductive health histories. These are not factors that are commonly captured in our brain aging and neurodegeneration cohorts. I'm hoping this is going to change in the coming years, but we really need to understand these critical health events that happen in half of our population. And there's a ton of incredibly exciting technologies that are now allowing us to capture a whole host of other biological pathways in vivo from biofluid samples, looking at transcriptomics, proteomics, metabolomics, all the omics. So in real time, we can start to disentangle some of these biologies that might be driving some of the sex differences. Lastly, I would just argue that understanding biological sex and neurodegenerative disease is really the first point in developing these precision care models for dementia prevention. This is just a really table stakes, and I think we can all do this. So with that, I want to just thank everyone that's involved in this work, particularly the participants and families that make this work possible, give me a job, and of course to the UCSF aging team, incredible colleagues and friends that I get to work with, as well as all of my collaborators. And I'm happy to take any questions.
Great. I have a pressing question. Is there a difference in the diagnostic criteria between men and women?
No.
This is like the biggest problem, right? Because if women start out with a higher verbal and better memory score, then there should be a smaller window to catch that early onset, right? Like the earlier onset. So I feel like that's your first, maybe that line you had is actually, you have the same line for males and females for diagnostic criteria, but maybe it should be higher for female. And then you catch them earlier and you can intervene earlier and get all these like sort of biomarkers at an earlier stage. But
yeah, no, and so, and we do have, we can norm for sex in some of our cognitive tests. So if we norm for sex on memory testing, you see this, the female advantage goes down, and then you can hopefully catch them earlier. And Erin Senderman's group has done a ton thinking about this as well. But I will say, not everyone norms for sex, and it's hard. It's still hard in the real world, I think, to capture this.
Yeah, because of course it's gonna be easier if you do have a diagnostic test that everybody's implementing, that it's the same score. But it seems to me that that's a missed opportunity to identify people who have this little, window where the treatment is most effective, right? Is like when you first start to have that cognitive decline. But women were like we're missing a bunch of them because we have the same line for male and female. But And then my second question, and then I'll let someone else ask you a question, is could the sex differences be totally driven by the SNAP-25 subgroup of women? Like, what is the percentage of women that have a SNAP-25? Because it seems like that is the one that's getting that cognitive advantage.
Yeah, I think it's a provocative question. I don't think biology is ever quite that simple. So my bet would be it's not just those individuals. But the polymorphism is quite common. I was just thinking on my drive in this morning. I don't recall what the prevalence is, but it's pretty common. So I do
It
think
like even maybe more were in
right.
that group than in the group that didn't have the SNAP-25 when you had that kind of rain cloud plot. But
yeah.
All right, there's one question we have from, we'll take, maybe this is the last one because I see we're already over noon. Thank you very much for this excellent talk. Could you please discuss how the interpretations of these findings might be influenced by sample bias? For example, could the sex differences in resilience patterns we observe be biased due to the differences in participant retention and the conclusions drawn from cross-sectional versus longitudinal data?
Yeah, I think that's absolutely fair. We should always be thinking about sampling bias when we go into our data. And it's pretty clear actually that women participate in research studies more often than men. And so we really need to think about this deeply. I guess the one thing I would point back to is that ultimate outcome of survival. So this is again across species. So mice, chimps, all types of species, women live longer than men. If you sort of anchor ourselves there and work our way backwards, I do think this is a real biological phenomenon, which makes it exciting to study. But yeah, maybe we're overcalling it in some of these longitudinal studies where we have an overrepresentation of women, especially women who are living longer.
Great, thank you. Okay. With that, I think we'll wrap it up since we're a bit over, but thank you so much. That was a brilliant talk. I loved it.
Really fascinating talk and research. Thank you so much.
Thanks,
And I think
guys.
next month we're skipping for August break, and then we'll be back in September with who is it, Jaren, that's giving?
We will have Judith in October, and then confirmed speaker, and in December, Shayla Shanmugan from.
Oh, perfect. Great. All right. So look forward to those. Bye, everyone.
Thank you. Bye.